Hello everyone, welcome to Pharma. I post videos on pharmacy related subject explained in a very simple and easy way. If you find my video useful for your studies, don't forget to like and share my video and subscribe to my channel. So today we will discuss on pharmacokinetics and various mathematical model for drug design, that is various pharmacokinetic mathematical model for drug designing. So it is the part 1 of this portion, that is pharmacokinetic mathematical model for drug design basically, because mathematical model for drug design also includes QSR. So that will be discussed in the part 2. So it is part 1 where we will discuss the pharmacokinetic models. So anyways, the brief introduction to pharmacokinetics has already been discussed in the previous video, that is last week's video. So you can just go and watch. It is in both Hindi and English explanation version. So you can just go and watch whichever you want to just feel comfortable in understanding. So anyways, so basically these portions I am discussing under computer application in pharmacy. So the portion which I am discussing, it is just beneficial for the topic under computer application in pharmacy, which is included in B Pharmacy second semester. So starting with the various, first of all, the various applications of this pharmacokinetic software in drug modeling and its analysis. So why this pharmacokinetics? Few softwares name I have already told in the last class, so that is last video. So why those are important, why these pharmacokinetic softwares are used and how they are helpful? So basically they are helpful in discovery of new medicine, then a particular candidate selection, that is out of various drug molecule, which one will be the best, that can be understood by knowing which of the particular drug molecules shows better pharmacokinetic result, that is absorption, distribution, metabolism, or interaction with our body. So next is also this pharmacokinetic softwares help us to characterize the safety of the new medicine, that is the particular new drug which we are inventing or which we are discovering, to know that whether it is safe or not, how much it is getting absorbed, whether it is crossing the toxicity level, that is maximum safety level, it is crossing or not, that is it is getting into our body and producing a toxicity level or not, and whether it is or if it is absorbing sufficiently to at least create a minimum value to produce therapeutic effect, that is whether it is at least crossing the minimum value or is it just not effective only. So to know that whether it is safe or not, or whether it is uh, better absorbed or not, and it is clearing out of our body by excretion or not, for all these things, that is basically the pharmacokinetic, which already I have told in a brief. I have explained what is pharmacokinetic in the previous video. So anyways, that part, to know that part, pharmacokinetic software is very helpful for discovering a new drug entity or modifying the dosage form of previously invented drug or previously marketed drug. So also to make a trial in the patient and know the efficiency, that is you test the medicine, or you trial the medicine, go for trial of medicine in patient, and after that the physiological record can be recorded, physiological studies can be recorded with help of various such pharmacokinetic softwares, that is how the medicine is going into our body and how our body is reacting with the drug, that how body is absorbing or distributing or metabolizing or excreting the particular drug. So that we can, for that we can connect particular physiological system of the body of the patient with this pharmacokinetic software, so that by this we can better understand that how it is behaving, how the drug is behaving inside our body, and then to determine the dose, that is to know how much sufficient dose we should give so that a particular dose can generate the required therapeutic level in our body. We should not just add or take that much dose that it crosses the maximum safety level, maximum therapeutic level, and it produces toxicity, and nor we should take the minimum amount that just it is not even producing the therapeutic effect also. So our purpose is that we should take any medicine, any dose medicine of any dose which can at least produce the therapeutic effect. That is there is a minimum level and a maximum level. There is for each and every drug which we take, there is a certain limitation of that drug's dose. That is, within that limitation only we should take. Out of the limitation it will be toxic, and under that limitation it will be of no use, it will have no action in our body. So anyways, for that particular criteria, to know the safety level and all, we can use this pharmacokinetic softwares. That is for drug discovery, selection of particular best candidate out of various candidates, then know the safety level of that particular candidate and how it is interacting with our body, then to conduct the trial by connecting the body of patient with the pharmacokinetic softwares, and then deciding the particular dose amount. So anyways, next, the various mathematical models. There's just a silly mistake by me, that is I have forgot to just change the heading, so please excuse that thing. It is not the introduction to drug information system, it is the introduction to various pharmacokinetic mathematical model basically. So anyways, so here it is. There are basically the major basic six pharmacokinetic mathematical models which are calculated manually also, and the various pharmacokinetic software use these mathematical models. The six which I will discuss now, that mathematical models are being used by various pharmacokinetic softwares to just calculate or estimate the effect of the drug, that is the pharmacokinetic effect of the drug, not the dynamic. So these are as such. First of all is the zero order model. Zero order model is being used for those types of medicines or those type of drugs which do not disaggregate in our body, and generally it releases very slowly. If we are preparing any such kind of dosage form which releases the drug in a very slow rate, that is, there is no effect of concentration in the rate of the reaction, that is from the starting point to the end point of the medicine's concentration decrement. That is, for example, if we are taking 500 mg of a tablet, and from the starting to the end, whether it is at the starting whether it is 500 mg, or at the end where it is just 5 mg left, if its rate of reaction is just 2 mg per second, from the starting when it is 500 mg to the end when it is just 5 mg left, then also the speed, the rate of degradation will remain as it is, that is 2 mg per second speed, or you can say the rate of the reaction, that is rate of degradation of the drug inside our body. So if this system is occurring, that is, it doesn't matter how much concentration is there. Like if that I will discuss in the second model, that is first order, where in the starting when that concentration is more, the drug is degrading in a speed way, that is the drug is degrading in a fast rate. Okay, and after that when the drug's concentration is decreasing continuously, so at that point when the concentration is decreased, likewise the speed of the reaction also decreases. Means more the concentration, more is the speed, less the concentration, less is the speed. So that is followed by first order, but it is not like that. It is zero order, where concentration doesn't matter. If the drug is from the starting point, it is releasing in a very slow rate, then those types of drug dosage forms are generally studied by zero order model. That is, if we are making a control release dosage form. Consider just we are making a control release dosage form in which no matter how much concentration of the drug you are taking, it will release its drug content in 2 mg per second speed only. So that particular type of medicines degradation or pharmacokinetic value is studied by zero order model. That is q naught by the equation is q naught minus qt is equals to k naught t, where q naught is the initial portion of the drug, initial amount of the drug, qt is the amount of the drug after particular time. That depends on us, that we want to read how much drug has been degraded after 1 hour or after 2 hour or after 10 hour. So that is the time, at particular time, when we are measuring the amount of drug which has been degraded, the amount of drug which is remaining. So qt is the amount of the drug which is remaining after time t. That can be any time, t can be any time under which we want to just evaluate the value of the drug left. K naught is the zero order release constant, so which can be calculated by subtracting the initial concentration minus the amount of drug after particular time. So if we will deduct the initial concentration from the amount left after particular time, we will know the rate constant. Because it is zero order, so the rate constant will be fixed, so we can know that at which rate the reaction is going on, the degradation is going on. So as I have told, application of zero order is to study or design coated type of medicine or transdermal medicine. In transdermal, the medicines are penetrating through our skin's layer. So through the skin's layer, no matter how much concentration you are applying over the skin, it will absorb in its fixed rate. That is, it depends on how much penetration power your medicine have. If your medicine's penetration power is fast, then it will penetrate the transdermal layers fastly. If its penetration power is slow, it will penetrate your skin layers slowly. So it doesn't depend on how much concentration of medicine you are taking, it just simply depends on the penetration power of the medicine through the skin's layers when we are applying it in a transdermal or injecting transdermally. There are various injectables also which we put transdermally actually. So anyways, in that case, or if we are preparing tablets which are coated tablets, so due to coating, it provides sustained release or control release of the drug. That is, in a fixed rate, the drug will release out of the coat. That is, there is a polymer coating over the medicine in the tablet, so that the medicine will penetrate and come out of the coating, polymer coating in that particular tablet. The drug will come out of the coating in a fixed rate. So next comes the first order model. So first order model, as I've already told, it depends on the concentration. That is, whether this equation can be for absorption or metabolism or excretion. For any type of pharmacokinetic factor, we can use this equation. For example, we want to know the excretion level. So whether the excretion is in first order or second order or zero order, for that we can use this model. Like if we know that particular medicine, when it is in high amount, it excretes, we just have estimated, we have guessed that the medicine, when it is in high amount, its excretion is fast. When it is in low amount, that is, gradually the medicine is coming out of our body through excretion. For example, excretion through urine, it is coming out of our body. After certain time, gradually its concentration will decrease in our blood. Then after the decrement of the concentration in our blood level, the rate of excretion also decreases. So such type of doses form a drug just simply follows first order reaction. That is, it depends on the pharmacokinetic effect parameter, depends on the concentration. When the concentration is high, the particular parameter is also high. When the concentration is low, the parameter is also low. Either it is in case of absorption or in case of excretion, and so on. So the equation for calculation can be Qt is equals to Q naught e to the power minus K1 T. So Qt is the amount which is released at particular time t, that is amount of drug released at time t, that is after time t how much drug is left, and Q naught is the initial concentration of the drug, the starting concentration of the drug which are taking. K1 is the first order rate constant. So anyways, this application is to study and design generally water soluble drug, because the drugs which are water soluble, which get solubilized in our blood easily, they are generally concentration dependent. That is, at the starting when the concentration is high, it will absorb easily, it will excrete easily from urine, etc. And when gradually the concentration is decreasing, the rate will also decrease. Then Higuchi model. Higuchi was the first scientist who created a mathematical model for for studying the drug release through matrix system. Matrix means polymeric matrix system, where various drugs are being coated inside a polymer, and when the coated polymer swells, the drug penetrates out of the polymer in a controlled way, that is in a fixed rate. That is, the drug is generally inside a polymer matrix, or you can say like a polymer bag is there. Inside that polymer bag there is medicine. It is not a tablet or it is not a capsule, it is just a polymeric bag inside which the medicine is being inserted either in solution form or in solid form. And these polymers are generally hydrated type of nature, having hydrated type of nature, that is it gets hydrated or swells when it comes to the contact of water, and likewise when it swells, it pushes pressure inside, that is the bag. For example, the polymeric bag is swelling, so when it will swell, the space inside that bag, polymeric bag, will decrease, and when the space inside the bag will decrease, the content which is inside that bag will come out automatically. So likewise the drug will come out of the polymeric bag through penetration through the polymers. That is, when the polymer is not swelling, the medicine is inside it, that is medicine is being locked inside the polymeric bag, polymeric matrix. But when the polymer is swelling, its pore size increases, and when the pores between these polymers increases, through that pores the drugs comes outside. So anyways, it is the matrix system which you can know in very details when you will study the noble drug delivery system, which in future upcoming videos I will start with noble drug delivery system also. So anyways, this model came into existence from 1961. The expression or the equation for this model for studying the matrix system is Q is equals to A to the power D, and then inside bracket it is 2C minus 2CS bracket close into CS into T. So Q is the amount of the drug that is released at time t, that is after time t, the amount of drug which is released from the matrix system. C is the initial concentration of the drug, and Q is the drug at time t. So here CS. So CS is the solubility of the drug solubility in matrix media, that is the power of the drug to get solubilized inside the polymeric media. Because when the polymers swell, so what happens? Polymers swell because water molecules or the physical fluid inside our body, it gets inserted into the polymer. Okay, that is why the polymer size increase, polymer molecules trap the water, so the polymers get swelled. And so what happens, that is the drug when it comes out of the polymer, or when the water goes into the polymer due to swelling, the water goes into the polymer, so water comes in contact with the drug, and likewise the amount of the drug which is dissolving in the water will come out of the polymer. That is, polymer is without water, a bag like structure. inside it there is medicine, that there is drug. When it goes into our body, the body's physiological fluid like water swells the polymer, and when the polymer bag is swelled, so what happens that swelling is happening because water is going inside the polymeric matrix. So when the polymer matrix is having the water molecules in its structure, so the so the drug which was inside that polymeric bag will come in contact with the physiological fluid or water like fluid. So the amount of drug which is dissolving in the water will able to come out of the polymer. If the drug is having low solubility, it will come in a slow rate through the polymer, okay, when the polymer is swelling. Other than that, it will not come. And if the solubility is high, the drug can come out of the polymer in a fast rate. So thus CS depends on the drug solubility inside the polymeric matrix, and then D is the diffusibility of the drug through the matrix. So two things is required. The drug should be soluble so that it can come out come out through the swelled polymer matrix. Also there should be diffusibility, means the drug should also have the power to diffuse, to penetrate also. That is, if the water is if the drug just in case you think that if water is not going inside the polymer, there can be various types of polymeric design. Various polymer matrix are having such type of design that water cannot go inside, just water can put pressure outside the polymer. That is, the polymer is not a swelling type of polymer, just the waters outside the polymer will put pressure to the polymer matrix, that is polymer bag. And when there is so much pressure, there is much osmotic pressure which is putting into the polymer bag. So the drug, because when you pinch something, when you squeeze something, what happens, like if you are putting pressure into a ointment tube, the ointment content will come out, like that happens. That is, if the polymer is not a swellable type of polymer, then the drug will come through penetration, not through swelling polymer. Just the drug have its ability that it can penetrate through, like in the cells there are various materials which go out or goes into the cell or comes out of the cell. So like that, the drug can also have the ability to penetrate or come out of the polymer. So there should be diffusibility power of the drug. So as I told, you will know it better way in the noble drug delivery system, and also I have explained this in Hindi also, so you can also go and listen this or understand in Hindi also. So anyways, the application, if we talk about the application, it is used to study or design low soluble drugs incorporated in a semi-solid or solid polymer matrix. As I already told, it is for studying the polymeric matrix based dosage forms. Now the Weibull model. The Weibull model, the fourth model, it describes the dissolution process. And what type of dissolution process? So basically I am explaining this in the application. So the equation for this type of model is m is equals to m naught inside bracket 1 minus e to the power minus small t minus capital T to the power b, and this whole term, that is minus small t minus capital T to the power b, is divided by a, and all this term is in exponential power form, where m is the amount of the drug which is being dissolved at particular time t, and then m naught is the initial, that is total amount of drug being released, or the initial amount of drug which is being released in your body, the starting amount of drug which has been released from the dosage form into your body, the initial concentration. T is the lag time, that is particular time which is required for a drug to come out of the dosage form. Okay, that is lag time, because here we are not talking about the initial concentration, we are talking about the initial concentration when the drug is released. Like you have taken a 500 mg paracetamol tablet. So from that tablet is the dosage form, and from that tablet, the time which is taken by the paracetamol drug to come out of the tablet or release out of the tablet will be the lag time. So capital T is the lag time for releasing of the drug from any kind of dosage form in which it is being loaded, and m naught will be the total amount of drug which is released after releasing the total amount of the drug that is available, and m is the available or the remaining drug after time t, that is total released from the released amount of drug, the amount which is left over after particular time t. A, where A is the scale parameter which describes the time dependence. So scale parameter, that is how you are taking the time, the consideration of time, that is how the particular drug is depending on the time, how particular drug is reacting along with time will be considered by A parameter, and B is the shape of dissolution curve progression, that is what type of dissolution curve it is creating. Likewise, exponential curve or parabola curve, like bell-shaped curve, or just a curved graph. Okay, so various types of curve. Sigmoid curve is S type of curve. So what type of curve means, when we are creating a graph of the amount of drug releasing pattern, how the drug is releasing particular time. So for that we have to take various value on various time interval, and we have to see according to various time how it is reacting. Like if in a bell curve, for example, like a school bell, a bell curve, it means the drug is releasing when at the starting point till the maximum time for particular certain, you think that for certain concentration, until 50% amount is releasing, it will release at a high rate. Okay, it will dissolute at a high rate as the time is being progressed. Like in zero time, the release is little getting fast, and then at time at 10 minute you will see little more fast dissolution. Then at 20 time, that is at when the time is getting 20 minutes, more fast. After 30 minutes, more and more fast. Then when after 30 minutes you will see that the concentration of the drug has reduced to 50%. After that, when after 10 minutes you will see the reading, that is total 30 minutes. After 30 minutes, when the concentration, you think that you assume that the concentration has decreased to 60%, so only 40% of the drug is remaining, you will see that the speed of dissolution is decreasing. That is, initially that will increase dissolution rate, and then after certain concentration the dissolution rate is decreasing. In that case, after a certain increment, when there is a decrease, after reaching a maximum value, when again the speed is decreasing, dissolution is decreasing, then you will get a a bell like curve or a bell like graph. So various type of graph, which type of graph is being created when you are studying the dissolution rate of the drug at certain time periods, at various time periods. So that is expressed by B. Like 1, generally 1 is for B value, 1 is for exponential curve, 2 is basically for sigmoid curve, and then 3 is, I think, for hyperbola. So there are various constant values given for various type of curve which is being created by the particular medicine. So anyways, the application is for comparing the release profile of the matrix. So here this type of the Weibull model is being studied for comparing the drug's dissolution or the drug's release pattern through the matrix system. So by using various types of polymer matrix, you can just compare that which polymer matrix will give better release pattern or better dissolution pattern. So we can select, so that we can select the best formula or best polymer for creating the matrix system. And then fifth comes Hixson-Crowell model. Hixson-Crowell model is generally studied for tablets or any solid dosage form in which the shape of the tablet or shape of the dosage form remain as it is, just its size or surface area decreases. Like in case of tablet, you can see the tablet, if you put a tablet or disprin for example in a water, you will see if you just not shaking, you will not disturb the tablet, you are just putting it in a water, you will see that with the progression of time, layer by layer of the tablet coming out, and it is being degraded or disintegrated in the water. That is the shape, the round shape of the tablet is being maintained till the tablet disintegrate completely. The tablet will exist in its own shape, just its surface will remove by layer, one by one as a layer, and the size will decrease but the shape will remain as it is. So for that type of dosage form, generally Hixson-Crowell model is being used. The equation is W naught, that is W naught to the power 1 by 3, that is under your cube root. Okay, under cube root W naught minus under cube root WT. So under cube root can also be written as W naught to the power 1 by 3 minus WT to the power 1 by 3 is equals to KST, where W naught is the initial concentration of the drug in the dosage form, in particular dosage form like tablets. WT is the remaining amount of the drug in the dosage form after time t. Ks is the constant which represents the surface volume relationship. That is, as I told, shape will remain as it is, just its surface will remove layer by layer with the progression of time, and if the surface is removing, then its volume will also decrease. So Ks represent the constant of surface volume relationship of the particular dosage form. So its application is to use, it is used to study and design the dosage forms like tablet, as I already mentioned, where the dissolution occurs in the plane parallel to the drug surface. That is, the drug surface will remove as a layer. Okay, a straight line like layer will be removing where the dimension, that is the size, okay, size will reduce, but the geometric shape or the geometric form will remain as it is till it disintegrate or dissolute completely. Then the last is Korsmeyer-Peppas model. This relationship helps to study the drug release from the polymeric system. So it is also for polymers. So there are three models for polymer matrix. That is, first of all, Hixson-Crowell, then it is for Weibull model, and then the third is this Korsmeyer-Peppas model. So the equation is mt by m infinity is equals to kt to the power n, where mt by m infinity is the fraction of drug releasing at time t. That is, it is a ratio. Okay, it is the ratio to just get the value of the drug which can release at particular time. So mt, basically mt is the amount of drug which can release at particular time t, and m infinity is the normal releasing amount of the drug which is which is fixed for infinite time, the releasing pattern. So mt by m naught will give the value of fractional value of drug release at particular time, and k is the release rate constant, the constant value with which the drug is being released. N is the release exponent, so t to the power n represents the power of time which is being taken by the drug to release. Okay, and talking about the application, so it is used to study the drug release from various modified dosage form. It is just not for the polymeric matrix only, it can be any dosage form like nanoparticles or any type of dosage form which uses polymers to modify its dosage form. If polymer is being used, then we can just use Korsmeyer-Peppas model to study its pharmacokinetic system. So thus it is useful for designing modified dosage forms. So these are the mathematical models which can be studied to know the pharmacokinetic characteristic of particular drugs, so that it can be helpful to us to design the drug, and by using pharmacokinetic model, not by calculation manually, we can just simply calculate those values to know the characteristic of pharmacokinetic characteristic of the drug or particular dosage form. So next we will talk about the QSAR mathematical model for drug design. For more videos you can go and check my channel. Hope that you have found my video useful. If yes, then press the bell icon to get the notifications. Thanks for watching and bye bye.